Welcome to the Neuroscience and Behavior chapter. In this chapter, we're going to cover the brain as a physical object and how it controls behavior in general. There are a number of basic rules regarding the structure of the brain, the layout of the overall nervous system, and how it manages behavior. Things that set parameters on what a human being can do and what a human being cannot do. And those are the things we're going to wind up focusing on. To get started with, we're going to take a look at the most basic building block of the nervous system, and that's called the neuron. The neuron is the proper name for a single individual cell in your nervous system. Generally, in everyday language, you talk about nerves or the nervous system, but technically, a nerve refers to a bundle of individual cells that essentially make a cable that run through your body. The proper name for one individual cell is a neuron. And the primary job of a neuron, at least with respect to psychology, is that it will transfer electricity through it, and it will be part of an information processing system that will manage your thoughts, feelings, behavior, or any other thing that is associated with human psychology. Now, the neuron has three basic parts to it, and this is going to be true regardless of the neuron you're looking at. So if you look at neurons in your brain or in your spinal cord, in your arm, in your leg, or in your pinky toe, they all have the same general characteristics, and that is these three anatomical parts: a soma, a dendrite, and an axon. Now, it's true that the structure of the cell itself may look a little bit different in different locations of your body and depending upon the specific function of the cell. Those are issues that we don't need to be concerned with in a general psychology class. What we're going to focus on are these three universal components of neurons that exist in every one of the cells in any location of your nervous system: brain, spinal cord, arm, leg, heart, anywhere. Every one of the neurons has each one of these three components. And we're going to look at them in detail in the order that they're presented here. First the soma, then the dendrite, and then the axon. And the reason we're covering them in this order is because the soma is the location where your genes are held. And so we'll have a brief discussion of genetic control of behavior. The dendrite and the axon essentially work together as part of the information processing system of your nervous system. The dendrite will always pull information into the cell from other cells, and your axon will always send information away to other cells. The soma is also called the cell body. So to be clear, these are two names that mean exactly the same thing. And the soma is the place inside of the cell that houses the nucleus, and the nucleus houses all of your genetic material. So the soma itself is always a swollen section of the cell. It's very easily identifiable when you look at a neuron underneath of a microscope, because the structure of a cell is essentially like a snake. If we flip back one slide, the soma is easily identified in this drawing. It's the swollen section. At one end of the cell, there are a bunch of dendritic branches, and at the opposite end of the cell, there are a bunch of axonic branches. So the cell itself is essentially one large tube with branches on one side and branches on the other side. If you find the section of the cell that is swollen, that is always the soma. And the soma is the place where your genetic material is always held inside the nucleus, as you can see here in this image. Inside the nucleus is the location where all your chromosomes are. So if we play the game that you have a single individual neuron on a microscopic slide and you're continuing to increase the magnification of the cell, at first you'll only see essentially a tiny little line segment, which essentially makes up the cell itself. As you zoom in closer and closer, you'll start to see that on each end of that line segment, you have a bunch of branches, dendritic branches on one side, axonic branches on the other side. As you zoom in even closer, you'll begin to see that one section of that strand actually is larger than the rest of it. That's definitely the location of the soma. If you focus on the soma and continue to zoom in on that, you'll eventually see the nucleus inside the soma. Now, the soma is swollen in part because the nucleus is there, but there's a lot of other organelles inside of the soma that have other purposes in processing proteins. So the soma is not swollen just because of the nucleus. There are a lot of other systems present there that require more space to be used. We will not be discussing all of the various organelles inside of the cell. We're going to selectively focus on the parts of the neuron that are related to controlling overt behavior in the organism. So for our purposes now, what we're going to do is continue to zoom in our hypothetical example on the nucleus. And as you do, one of the first things you'll start to see is that inside of the nucleus, there are a variety of oddly shaped X's floating around inside of the nucleus. Those X's are actually your chromosomes. You're going to have 46 total chromosomes altogether. Now that's actually 23 pairs of chromosomes, giving you a total of 46. And the X's that you're looking at are actually the pair of chromosomes. So you have 2 chromosome number 1, and that pair of chromosome number 1 are connected together at the very center by something called a centromere. And because the chromosome itself is relatively flexible and it's floating in liquid, the 2 flexible tubes that are connected together at the very center essentially cause an X shape, and that's why your chromosomes look like an X. Now, if you were to take one of those pairs of chromosomes, pull it out, and snap off one of the chromosomes and throw it away, you would have essentially one flexible tube hanging. That would be a single chromosome of the one that you had selected. So let's say it's chromosome number 1. That chromosome, being relatively short and fat, is actually made from a very long string of DNA, the double helix that you're already familiar with. And so if you try to imagine in a metaphorical sense that you have a 10-foot length of sewing thread, but what you do is you take that sewing thread and you fold it up very, very carefully. So instead of being very, very thin, only the width of the sewing thread itself, and being 10 feet long, you fold it and fold it and fold it and fold it so that it becomes 1 inch thick, but only 3 inches long. That's essentially the way the chromosome is set up. It is a long string of a very, very thin string of DNA folded up so that its length has gotten much shorter, but consequently has become much thicker. So if you could take a chromosome and stretch it out, that very, very tiny chromosome would produce a very long string. And upon looking at that string, you'd be down at the molecular level because you are at the DNA. The DNA is, of course, the instructions for building all things that will make you. Now, along this string, you will have various sections that will be each individual gene. So you'll have in a hypothetical sense, a 5-inch section that produces skin cells, and then a 17-inch section that will give you your eye color, and then a 3-inch section that will give you your hair color, and so on and so forth down the line. And in fact, of course, as you might imagine, genes that are going to be relevant to each other will be more likely near each other in the chromosome, but you have along this string a biochemical marker that identifies the beginning and the end of any one particular gene. So in the broader story of the biochemical machinery that operates your genetic, there will be biochemical markers that the molecules will look for, and then it will begin reading the code for the DNA, and then it will reach a stopping marker, so then it will stop reading the code for the DNA at that one location, so that you'll pull the information from one particular gene off and then use that to start making copies of proteins that will generate those particular cells or whatever function is actually going to be generated from that DNA. Now let's consider this general question. This general question oftentimes is related to police shows that you see on television. It's a common theme that you're probably very familiar with, but I'm extending it into this story here. All of your genes are located in every neuron within your nervous system. Is that true or false? And that is true. The connection from this question to cop shows is always that every time there's a murder, the police look for DNA, and they can find DNA in any cell. They can find it in saliva or hair cells, skin cells, anything that a person essentially does or wherever they go. A single hair may fall off their head. They may leave some skin cells behind. And of course, in a police show, being overly dramatic, you can always find this DNA, scoop it up, and analyze it to be able to get the genetic fingerprint of the murderer. And that general idea is essentially true, but it also extends to all of the cells inside of your body. And in particular here, we're referring to your nervous system cells, your neurons. Every neuron that we talk about has a nucleus, and every nucleus has all of your DNA in it. So that's every single neural cell inside of your body has all of your genes in it. Now, the story of how your genes are activated is somewhat difficult, or really probably more a lengthy story as opposed to being difficult. And so I'm going to show you a short animated movie here to give you an idea about how the processes of transcription and translation occur. For our purposes in this general psychology class, the terms transcription and translation simply refer to the activity of collecting the information from a gene inside the nucleus, being called transcription, and then when that information exits the nucleus but is still inside of the cell, it's then referred to as translation. So transcription inside the nucleus, translation outside the nucleus. These are common terms related to neuroscience that any modern psychology student should be familiar with, but beyond that aspect of it, we're not going to delve further into the biochemical processes that occur. If you want to do that, I would recommend taking a genetics class. So let's get started with this particular animation.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecule called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
That video does a nice job of representing how your genes are accessed and what they actually do. In fact, one of the basic rules of genetics, especially with respect to psychology, is this. Your DNA has one job, and that job is to generate proteins. And as you saw in that video, the proteins are created by stringing together a sequence of amino acids. Then that sequence of amino acids is folded up, and then it is either used inside the cell itself or it is ejected from the cell for our purposes in psychology, likely as a hormone into the surrounding blood supply where it gets washed away. So there are two general pathways for the production of protein in a cell that's related to relevance in psychology. Either the protein generated stays in the cell itself and it will become a neurotransmitter. And that may be a term you're familiar with, but a neurotransmitter is a chemical signal that will work in a very local area between one cell and literally the next cell next to it. The alternative is a protein is generated and it is ejected into the blood supply that surrounds the cell itself. So in many cases, there are very small capillaries, which are the smallest branches of your circulatory system, that run along the side of these cells. And so when the cell generates a protein, it can eject it through the cell membrane into the blood supply where that substance will then just float off. In a scenario like that, that substance would then be referred to as a hormone. And that hormone will simply go wherever it goes based upon its chemical properties. So it doesn't know to go to your foot or your kidney or your liver or to your brain. It just washes away. Everything we're doing here is just operating off of basic chemical principles. So typically what you will wind up seeing is if a hormone is needed, a great number more molecules of the hormone will be created and pushed out into the blood supply, where only a small number of those molecules will actually reach the location in the body where they will actually have an effect. The rest of them will just wash around in your blood supply, get stuck in fat, or do something else that's largely irrelevant. Now, when we talk about genes and cellular signaling, what we're really doing is talking about these two basic ideas that I've been talking about. Genes create hormones. The hormones get released from the cell into the surrounding blood supply. Two of the most common areas in your brain, specifically, that release hormones are your hypothalamus and your pituitary gland. And in fact, sometimes that's referred to as the hypothalamic pituitary axis. It is essentially an area just above your nasal cavity at the lower portion of your brain in an area called the temporal lobe, where the hypothalamus and pituitary exist. In that area, there are a lot of capillaries, so when the hormones are generated, just as you saw in the video, those hormones will be ejected into the surrounding blood supply and then get washed away. Now, the hormones that are generated in your hypothalamus and your pituitary and in other locations of your body have a wide range of influences on your behavior. Hormones are not just things that occur for short periods of time and influence your behavior and then simply turn off. But your behavior throughout your entire life is actually modified by the presence of hormones that can be determined by their concentration in your blood supply. In fact, this is a short list, some of the things in which hormones modify on a regular basis. Feeding. You've all heard the story that when you eat food, the food fills your stomach, and as your stomach becomes distended, that is a signal back to your brain that you should stop eating. And that's absolutely correct. However, in the story of feeding and the feedback system from your gastrointestinal tract back to your brain involves a lot of chemical signaling as well. And in fact, as food gets into your stomach, the content of the food is either fat or protein, as it is in your stomach or in your intestines, causes a release of different types of hormones from your gastrointestinal tract into the blood supply, which washes around inside your body, and some small proportion of it, as it washes through your blood supply, will get back to sites in your brain which will serve as a feedback mechanism from your intestines back to your brain, telling you that you're processing fat and protein and that you should stop eating. These signals, of course, take a longer time to turn on, and they run for a longer period afterward, as opposed to stomach distension, which is the more simplistic story that people generally talk about. Now, if the hormonal feedback system from your gastrointestinal tract back to your brain is modified by increasing or decreasing the amount of signal, you could cause a person to become either anorexic or obese. So, for example, if there's a secondary disease process that is affecting the intestines, which is corrupting the feedback mechanism to turn off the brain signals indicating hunger, you could have a person not feel full and just continue eating. And as they eat more and eat more, they of course will begin to gain weight, and then they will wind up obese. And that will be because of this feedback mechanism not working properly. If in a different process you have excessive signaling coming from your intestines, always turning the signals off, you could make it so that a person is less hungry and eats less when they do eat, so that they begin to lose weight and ultimately be identified as anorexic. So obesity or anorexia may show up as mental health concerns depending upon what's actually happening with a person in their life. But as a good therapist, you have to rule out that there might be a secondary disease process in the intestines or in some other location of the body, which is actually corrupting the feedback mechanism and causing what appears to be a symptom of a mental illness. And these things have to be cared for very carefully, because if you believe a person's anorexic because of their individual psychological characteristics and their relationship with their parents and their life experience and so forth, and you start trying to treat them in therapy for these things, however, the anorexia is caused by a secondary disease process corrupting a feedback mechanism from their intestines, then you're essentially going to allow that disease process to carry on because you've misdiagnosed them and you're treating the wrong thing, so the patient is only going to get worse. So in many cases, as we'll see much later in the semester, some forms of psychological disorders can actually be caused by straightforward medical problems, and as a result, a good psychological therapist has to send their patient for a medical checkup before they focus on the psychological characteristics of treatment, because the medical aspects have to be ruled out before you can focus on the psychological ones. Emotions are also managed by your hormonal state. The fact that you experience happiness or sadness and oftentimes experience a lengthy period of an emotional state as a result of one specific event in the environment is oftentimes driven by hormonal activity. So for example, if you found out today that you won $40 million in the lottery, you very likely will feel happy for hours. That may not sound like an odd thing. Of course, anybody would be happy for a very long time after winning $40 million. But from the standpoint of the mechanisms of psychological processing, there actually is an odd event occurring, something that would draw your attention to it. Upon realizing that you won $40 million, you should be happy at that moment, and then the happiness should immediately subside. The fact that you're happy for many hours afterward indicates that the happiness is caused not specifically by the external event, seeing the winning lottery ticket, but rather the lottery ticket caused an emotional event that is driven by the release of hormones that carries on over the course of hours, which puts the person in a good mood. So that lengthy emotional experience of happiness that takes place over the course of hours is biochemically being driven by a hormonal release. It was initially activated by the presence of the winning lottery ticket, but then the pleasure over the course of hours is driven by a hormonal activity. If the hormones in this case for a certain person are corrupted by a secondary disease process, then you would see what might seem like a very odd event. The person would see the winning lottery ticket, they would feel happy, and then they would go back to their normal emotional state 5 minutes later. This same type of process occurs for fear and for anger when you imagine something like failing an exam and being angry hours later still. A lot of that actually is engaging hormonal systems that drive the emotional experience for a longer period of time. Sexual responsiveness occurs in a variety of different ways, but one of the simplest ways to actually identify this as a process is the behavior of individuals pre-puberty and post-puberty. And probably one of the easiest ways to identify this is the common presence when you talk to children. If you say to a bunch of 7-year-old boys, hey, you're going to go to a girl's birthday party, and then the 7-year-old boys might say, ew, girls. But 10 years later, when you tell them, hey, at age 17, you're going to go to a girl's birthday party, they might say, yay, girls. And what happened between that time? Puberty happened between that time. There was a release of hormones which has completely changed the pattern of behavior of the individual. So around the time of puberty, the concentrations of hormones that are in your blood supply are managing your behavior. So as you might imagine, in a broader story, one of the things that this oftentimes gets connected to is the reduction in testosterone in men at later years. So oftentimes by the time a person's around age 50 or so, their concentration of testosterone in their body is starting to reduce through natural processes. That oftentimes reduces their interest in romance, and that's in part where you start to see commercials for things like Viagra, as well as these health centers that will give you testosterone injections and tell you that your life will be fuller as a result of the testosterone injections. What they're really saying is, if we give you testosterone, you'll have more interest in romance, and then you can have more sex. That's essentially what's being stated, but they say it in a very polite way on the commercials. But what's really happening is the arc of concentration of testosterone in your body over the course of your life increases quite a bit after puberty and then slowly drops off toward the later decades of your life. Wakefulness is another one that influences your activity on a daily basis, but you may not be aware of it. The amount of sunlight you get exposed to during the course of the day influences hormonal release, which makes it more or less likely that you will feel awake or tired. And in fact, when less sunlight is going into your eye, you will be more likely to become tired. And this, of course, might seem like a natural biological process developed over evolution. When the sun sets and it's dark, human beings don't see so well, and so they huddle together essentially in their villages and become more quiet. It's sleepy time. And then when the sun rises and you can see better, you get up and you start to move around and become more active. That pattern is driven on a daily basis in part through hormonal activity, because the amount of sunlight that goes into your eye does not just process vision, but part of the light that goes into your eye is not used for vision. It actually takes a separate path from your eye to your hypothalamic pituitary axis and causes a change in release in hormones, which makes it so that you're more awake. So these are just a small number of ways in which hormones manage behavior. And then since the 1940s, there has been a field of study called behavioral endocrinology. Endocrinology is the medical term referring to the study of hormones. And anytime you take the word behavioral and use it as an adjective for any other field, like behavioral endocrinology or behavioral genetics, you always are talking about the mixture of psychology with that other field. The field of behavioral endocrinology is the study of how hormones manage behavior. And that's a very important field to evaluate because, as I've mentioned, medical processes that interfere with natural hormonal feedback systems inside your body can cause an individual to develop a psychological disorder. And that psychological disorder is really driven by a medical cause, not a psychological one. And so you have to be very careful when you're evaluating an individual so that they are treated by the proper process. Now let's ask a question. Which of the following psychiatric disorders might not be controlled by a hormonal imbalance? And the correct answer there is E. All of the above may be affected by hormones. Undereating, which was anorexia, aggression, so that the area of the brain that controls aggression, which is in part the amygdala, which is an area of the brain we'll look at later in this chapter, can be stimulated from various reasons. One common one that you do see is that sometimes an individual who is mild-mannered and polite to individuals, not aggressive, may start to develop an anger problem. And that anger problem eventually will be found to be caused by a cancerous tumor near the amygdala. So that as the tumor in the person's brain grows, it physically pushes on the amygdala. As the amygdala is physically pushed on, it will generate more electricity in that area of the brain, driving more aggressive activity. If the cancer is then addressed and the cancerous tumor is eliminated by surgery or through radiation therapy or other types of things, and that cancerous tumor shrinks, the amygdala is no longer pressed upon, and then the aggressive behavior will subside. Other things like this also occur for hypersexuality or hyposexuality as well, and sleep disorders. Different types of medical disorders can cause an activation or deactivation of hormonal feedback systems, which will cause a psychological disorder. But again, it's always important to distinguish between whether or not that individual has the disorder through a medical cause, which you would call a medical etiology, or a psychological cause, because either the medical treatment or the psychological treatment will be completely different treatments. This leads us to the next two sections of the cell, the dendrites and the axons. The dendrites are the branches of the neuron that always lead to the soma itself. In this image on the screen, you can readily identify the dendrites because they are always pointing at the soma. The soma was easy to identify because it has the nucleus in it. The branches of the cell that lead directly to the soma are always going to be the dendrites. And the job of the dendrites is to bring charged particles into the neuron. And they're bringing charged particles into the neuron because in the sequence of cells connected from cell to cell to cell to cell, some other cell, not drawn in this image, has stimulated the tip of the dendrite. And the job of the dendrite is to let charged particles into it, and those charged particles will run up the dendrite and into the soma. This is just part of the entire sequence of events that we're actually going to talk about that describe the process by which the cell is going to be sending information around inside of your nervous system. Now, these cells are going to bring charged particles into them through the dendrites. The charged particles will reach the soma. Now notice that the dendrites are a very thin tube and they reach this large voluminous soma. So there's only a short period of time, actually 1 millisecond, through which the charged particles can reach the soma before they begin to diffuse out into that larger area. If they have not diffused into the larger area of the soma, they will have an influence by changing the chemical makeup of the cell membrane itself so that it will generate electricity. And to understand that, we have to continue on with the story. So essentially, I'm just teasing you a little bit with some of the information, but I can't just go through the entire process. We have to work through this step by step to understand what's actually happening. As the charged particles come in through the dendrite and reach the soma, they have to cross a threshold of a positive 10 millivolts. Now, positive 10 millivolts is the threshold before the cell itself will generate electricity. We have not talked about the cell generating electricity yet. We're only talking about some of the basic chemical properties that are going to cause electricity inside the cell itself. Note that positive 10 millivolts is a very important number to remember. The sign on the millivolts is going to be very important when you talk about the electrical activity inside the nervous system, because there are things like negative 10 millivolts under some circumstances that can occur and positive 10 millivolts in other circumstances. So the sign is always going to be important to keep in mind. When the cell generates an electrical signal, it'll always start at the very beginning of the axon. And that section of the axon is referred to as the axon hillock. It's simply the name for the very beginning of the axon, just off of the soma itself. Now let's take a look at a question here that's related to this activity. Each neuron only creates a new signal if it's stimulated enough from its surrounding cells. That is absolutely true. And in fact, to understand this a little bit better, what we'll do is take a look at this image again. And you can see here the dendrites along the top of the image should all be connected to a wide variety of other cells in a much larger pathway connecting the cells from cell to cell to cell to cell. And of those surrounding cells that are not drawn in this image, each one of those surrounding cells will stimulate the tips of the dendrites themselves. And if enough of the dendrites are sending positively charged ions into the soma, then the cell may cross its positive 10 millivolt threshold within 1 millisecond, and then this cell itself will generate an electrical signal at the axon hillock. Now, the electrical signal only exists in the axon itself, and this is an important part to remember. There is electricity in your nervous system. We talked about that back in Chapter 1, because back in 1745, the Leyden jars were invented, and then Galvani was associated with the discovery of the presence of electricity inside of the nervous system. Now, it had been largely assumed that the electricity just traveled through the nervous system, essentially the way electricity travels through wire. But that's not actually the way it works. As we're going to wind up saying, there is charged particles that run through the dendrites, then electricity that runs down the axon, and then there will be a chemical signaling process between cells. So when we talk about information passing through the nervous system, we're really talking about a sequence of 3 separate biochemical events that always occur in the same sequence. Charged particles through a dendrite, electricity down an axon, and then a chemical signaling process between cells. So we have to get through the entire process to actually understand the cycle of events that passes information through the nervous system. Right now, we're just focused on the second component of this three-term process, and that is the electricity traveling through the axon. Now, the electricity that travels through the axon always has the same strength, and that is a positive 40 millivolts. Now, note in general, in terms of studying, we are starting to talk about different voltages. Positive 10 millivolts is a threshold to generate electricity, but when the electricity is generated, it is always positive 40 millivolts. It's important to keep those two ideas separate in your head. Now, the proper name for electricity in your nervous system is an action potential. So you can talk about electricity, or more technically, action potential. And action potentials are always the same size. They are always positive 40 millivolts. Now, the speed of an action potential down an axon moves at a rate dependent upon the diameter of the axon and whether or not there's an insulating property called myelin on the axon itself. The estimate that we saw from Chapter 1, that electricity travels through your body at 90 feet per second, is essentially a general estimate for most cells. So that back in the 1850s, when Helmholtz was evaluating how quickly electricity traveled through the nervous system, he did not realize that he was accumulating the speeds of a variety of different cells together and estimating an average of about 90 feet per second. But that's essentially how quickly most signals travel through. If you look very closely, though, some cells, if they have a larger diameter and an insulating body called myelin wrapped around the axon itself, the signal can travel upwards of about 240 feet per second. So much faster than the 90 feet per second that we were talking about before. And in fact, in later classes, if you take a more advanced biology class or more advanced psychobiology class, one of the things that you'll see is that the brain uses these different speeds of transmission in circuits to send signals sooner or later so that the signaling system can coordinate itself. Now, when a cell fires, it either fires as positive 40 millivolts or it does not fire. That's called the all or none law. The cell either fires or it doesn't fire. No extra strong firing. Essentially, a cell firing or generating an action potential produces a positive 40 millivolt signal, and that's it. You don't see positive 30 millivolt signals or positive 20 millivolt signals, and you don't see positive 60 millivolt signals either. The size of the signal is always the same. And that's why it's called the all or none law. One of the interesting things about this is that when you consider all of the various thoughts and different patterns of behavior that you can have, they are ultimately driven by the exact same type of electrical signal in any one cell. It's always a positive 40 millivolt signal. And every one of those signals always follows its way down the axon in a punctate process. So the signal, the electrical signal, travels down the axon itself. It has a front end and a back end, and it travels down the axon. So every thought you ever have is essentially coming down to a basic stream of electrical signals that are either on or off. It's a dichotomous, binary process. Like when you talk about ones and zeros inside of a computer, you have a very analogous system inside your brain that is electrical signal on or electrical signal off. Now let's ask two questions about some of the material we've been covering. Action potentials within a neuron travel from the dendrite to the axon and across neurons from axon to dendrite. Take a moment, stop the movie, And think about this very carefully, and I'll give you the answer in a moment. The correct answer there is false. Action potentials do not travel from the dendrite to the axon. Charged particles travel up the dendrite, and then the action potential travels down the axon itself. So the first part of that statement is not correct. The second part of the statement is also incorrect. The action potential does not travel across neurons from axon to dendrite. I mentioned earlier that there is a chemical signaling process that interacts between the cells. That process is going to be called neurotransmission. The next question I have for you is this one. Which of the following is the resting and active voltages of a cell? Well, I know in this case I did not tell you anything about the resting voltage of a cell. Hopefully, you've already checked the textbook. The correct answer there is B, minus 70 millivolts and positive 40 millivolts. If you were to try to deduce this answer, the active voltage of a cell, when a cell has an action potential in it, that cell will have a positive 40 millivolt signal in it, which means that either answer B or answer D would have to be correct. To know that the resting potential of the cell sits at minus 70 millivolts, you would have had to have already checked the textbook or known from some other reason. Now, the cell itself sits at minus 70 millivolts, and that may seem rather odd if you're not very familiar with physics or electricity. How can a cell sit at minus 70 if it's not doing anything? Well, it operates the same way as a battery. If you have a D-cell battery that you would put into a radio, for example, that battery says it has 1.5 volts. What are the volts? If you pick up the battery, you're not being electrocuted. The voltage is a potential for energy to come out of the battery itself. Electrons will come out of the negative side of the battery, run through the electrical circuit of the radio itself, and then those electrons will come into the positive side of the battery. Essentially, the battery itself has two compartments. One side that has an excess of electrons, and the other side of it, which is physically isolated from the opposite side of the battery, can receive electrons. So when you place the battery into an electric circuit and you close all the switches, the electricity, which is essentially the electrons, travel out the negative side of the battery, through the circuit, and then into the positive side. You have a similar process taking place here with respect to your cells. A neuron sits at minus 70 simply because it has many more negatively charged ions inside of the cell than it does outside the cell. And in fact, the ratio of the negatively charged ions inside the cell relative to the outside of the cell is going to be minus 70 millivolts. And that's where you get the baseline or resting state of a cell as minus 70. Note also that when the cell fires, you get an action potential and the voltage raises to a positive 40 millivolts. You have actually changed 110 millivolts from minus 70 up to positive 40. Remember, I had already mentioned the sign on the voltages is very important. You are operating on a number line here from a negative side to a positive side. So to go from minus 70 millivolts up to positive 40, you're going to travel a total 110 millivolts. And you're going to travel in the positive direction.